Hi, welcome back to Discovering Patterns in the Microbiome. In this video, we will be discussing compositionality and how it can affect biological conclusions in microbiome data. So here's a very nice example from this great technical report written by David Lovell a few years ago, showing what compositional data can look like when you know what's happening in the actual abundance and when you're restricted to the compositional relative abundance. So compositional just means that the data are measured in fractions. You don't know how much of everything there actually was, just what percent of each feature made up the total. So, here's a great example where you're shown several different features. So let's pretend these are species. And you have a whole bunch of species, this red, brown, green, turquoise, blue, and then pink. And as you look at these four different treatments going across the top row, the only thing that's changing is that this dominant bug, the pink bug, is decreasing from treatment to treatment as you move across. The other ones stay exactly the same. So when you convert this to relative abundances, it actually changes the way that you would interpret the data. If you look at the first plot, it looks the same, but when the pink species decreases, the fraction of the community of that species decreases, which means the fraction of the other bugs increases, and so all of those bugs go up at the same time that one's going down, even though we know in the absolute abundances that didn't happen. So as pink goes down lower and lower and lower, you see all of the other bugs going up and up and up together. And this can make you conclude several different things. You could think that treatment F, that's the last column here, that treatment F is causing these other minor members to increase relative to treatment C, the first column. And you could also infer that, firstly, the pink bug is anti-correlated with all of the other bugs, because as it goes down, the other ones go up. And you would also infer that the minor bugs are positively correlated with each other, because they're all going up and down together. So when in fact, there's actually no correlation in the data, and only the pink bug is affected by treatment. So this is actually a very real problem in microbiome data, at least for now. And there are a number of effects. So here's another example. Let's say you have a healthy and diseased treatment. These are the fractions of bugs you've observed in each treatment. And what you conclude if you look at these fractions is that, again, the last species went from 0.2 to 0.6. So you'd think that species is up in disease. And then the other species all went down from healthy to disease. So you think that those are higher and healthy. But the problem is that you don't know the absolute abundance of each community that generated those fractions. And this is a real number that we don't have. This number is called the basis for each community. So here are some different bases that could explain that data. Let's imagine that the healthy subject had a basis of 100. Then that means that the fractions have to sum, or the counts have to sum to 100. So that means that this community had 10 of the first bug, 10 of the next bug, 60 of the next bug, 20 of the last bug. Now, let's assume that if we assume that the disease sample had a basis of 100, then the conclusions would be the same as they were with the relative abundances, because the bases are the same, it didn't really matter. But if you instead assume that the disease sample had a basis of 200, so the total count, this could be number of cells per gram of stool, for example. If the total basis was 200, then the disease subject would have actual abundances of 10, 10, 60, 120. In which case, if you compare healthy to disease 2, again, there's no change in the first three bugs, and only the last bug went up. So so this is the term that we're going to use throughout the rest of the video to describe the total abundance, the actual absolute abundance of bugs in each sample. The term is basis. Now, there's a very nice example of this that was just published in PNAS, showing compositional artifacts in practice and a way to deal with them. This was in a study of diurnal oscillations. So the authors took mice and gave them a standard diet and then tracked their microbiota at different hours of the day, and saw that there were these consistent cycles of different bugs throughout the day where the bug would increase for a while and then decrease. And what they show in their figure, one of their main figures, is the relative abundance of Bacteroides. And here you see that Bacteroides is high in the middle of the night, it goes down in the morning, and then it has a peak in the middle of the next night, and then it goes down, and then it comes up for another peak. And then if you look at Firmicutes, the opposite happens. So it's low in the middle of the night, goes up in the day, goes down in the middle of the night, goes up and goes down again. However, in this study, they were also able to measure total abundance using universal quantitative PCR primers for the 16S ribosomal RNA. So it's basically a primer that matches most 16S genes, ribosomal genes, and quantifies the number of copies of those genes per gram of feces. And so here they have a plot also of the total load, and you can see it's high in the middle of the night, it goes down a little bit, goes up again, then goes down a little bit. And so now you should be starting to wonder if there's something else going on here, because firstly, the Bacteroides signal and the Firmicutes signal are perfectly anti-correlated. So whenever Bacteroides goes up, Firmicutes goes down at exactly the same amount at exactly the same time. And then the Bacteroides is also almost perfectly correlated with the total bacterial load. So what they did was to multiply the fractions, that's what's shown in the relative abundance, by the total load in each sample. And now they have absolute counts rather than relative abundances. And then this is the transformed data. So it turns out that Bacteroides is actually much more prevalent, about 10 times as prevalent as Firmicutes in these samples. And its signal doesn't change very much. It's the dominant bug, so it was taking up a fairly large fraction of the community no matter what the total abundances were. And you can see that it follows the same pattern. But Firmicutes, you can see here, is actually much lower abundance on average, and it has a completely different pattern when you look at its predicted or projected absolute abundance than when you look at the relative abundance. So Firmicutes no longer negatively correlated with Bacteroides. In fact, if anything, it might be a tiny bit positively correlated, or at least it has a little bit of a bump in a couple of the places where Bacteroides does. So why did this happen? Bacteroides is more dominant. Let's say Bacteroides is 5 times as abundant as Firmicutes. If Bacteroides doubles in abundance, and Firmicutes, let's say hypothetically, also doubles in abundance, the amount of increase in Bacteroides was much larger because it started with a large amount. And so it's taking up the fraction of the community taken up by Bacteroides just increased a lot more, and actually causes the apparent fraction of Firmicutes to go down. And so this compositionality, if you're not careful about dealing with it like these authors were, can not only cause, it can not only add noise to your data, but can actually cause you to infer opposite conclusions, which is kind of scary. There are ways to get around this. So I just showed you one example. If you do qPCR with universal primers for your bugs, then you can just multiply the relative abundances by the total load. This works, it's cheap, it's easy to do. It's a little tricky to know how relevant it is in certain environments. If you're dealing with stool, for example, how do you compare total load in a diarrhea sample to a constipation sample? If someone has diarrhea, then there might just be a very diluted signal there, and so it's tricky. It's tricky to know. Maybe you should be tracking like the total load of fecal material produced by that person within 24 hours and normalize to that. A reasonable way is probably just to convert the units to cells, number of bacterial cells per gram of dry fecal weight. So you would lyophilize the feces or the fecal sample to get the weight of it, and then you'd normalize to that. There are other approaches. So another thing you can do is just spike in a known quantity of a bug that you're pretty sure is not in the community. And you could do this prior to extraction by spiking in actual cells of that bug, or you could do it after extraction by spiking in the DNA. It probably makes sense to do prior to extraction at least, but maybe even both of them with different types of bugs. And here are a couple references where that's actually been shown to work. However, most people aren't doing either of these things yet, and there's a lot of data out there that still don't have total abundances known, and we want to be able to analyze it. It's really hard to get rid of that problem where you don't know if the dominant bug went up in a treatment and all the other bugs stayed the same, or if it went up and they actually went down. So that's a little bit tricky to figure out. But there has been some very successful work in estimating correlation between bugs. So I mentioned in the first graphical example that you would infer that the pink bug was negatively correlated with the other bugs, even though they weren't changing. And you'd infer that the other bugs were positively correlated with each other because they were all going up together. When, again, in fact, there was absolutely no correlation. So what you want is basically the smallest number of correlations, or the most sparse representation of the correlation graph or correlation network that would explain the abundances, relative abundances that you see, given the fact that it's compositional data. Here's one of the tools for this, is a nice tool called Spark, and I'm showing you a comparison from that paper of 3 different body types. So this is vaginal samples, behind the ear and inside the cheek, and what we're showing is the correlation network. So everywhere that there's a green line is a positive correlation and a red line is a negative correlation. Between all of the OTUs on the outside of these circles, when using just raw Pearson correlation to test. And so you see a very dense correlation network here. If you shuffle the Pearson correlation, then, you shuffle the data and redo Pearson correlation, most of those associations go away. And then when you run Spark, which cleverly is trying to minimize the number of correlations that explain the data, you get even fewer actual correlations. So it dramatically reduces spurious correlations that are found when just looking directly at the relative abundance data. And the way that it does this is fairly clever and is built on a lot of theory that was published in the macroecology literature over the years, or not necessarily macroecology, but in high dimensional analysis. And the underlying premise is that when you take the ratio of 2 of your species, even if it's the fractions of them, you take the ratio of their fractions, their relative abundances, then that's the same as the ratio of their total abundances, because their total abundances to get the relative abundance had to have been divided by the same denominator. So you take the ratio, you take the log because it has some nice mathematical properties, and now we have this new quantity, Yij, which is the log of the ratio between xi, or species I, and species j. Now, the other interesting observation that they relied upon was the fact that if two species are highly correlated, then the variance of their ratio should be 0, or close to 0. And let's think about that. So let's say two bugs have these abundances, so their ratio of this one to that one is 2, for example. And then they both go up together, perfectly correlated, their ratio will still be 2. They go down together in another sample, then they go up, they go down. If they're perfectly correlated, then their ratio stays exactly the same and there's no variance in the ratio. So now they define a new term, Tij, which is the variance of the ratio. In fact, the variance of the log of the ratio. Then they do a little bit of other algebra and show that the same Tij is, it can be defined in terms of the variance of the absolute abundances of species I plus the variance of, or the squared variance, plus the squared variance of the absolute abundances of species J, minus this other term. And this term has the correlation in it. So what they would like to do is find all these values. They basically want to know the correlation term rho ij, but they have too many variables to solve for. So in these equations, for every pair of variables, there's an equation, which is n times n minus 1 over 2 equations, and there are n times n minus 1 over 2 rho ij variables, plus the wi and the wj variables. So that's too many variables to solve for. Instead, what they do is they start with the assumption that that most of the bugs are not correlated, so they set the rho ij's to 0, then easily solve for the variances, these w terms or omega terms, and then iteratively go back and figure out, if they assume that those are correct, what should the rho ij terms be, and then pick a few of those that they think are real, like the most correlated bugs, remove them, set the rest of them to 0, repeat, and they keep doing that a few times, and they figure out when to stop doing it. So that was the interesting insight that they had, and it works quite well. As you saw from that plot, it really dramatically reduces the number of predicted correlations. There has been a follow-up study on this, another tool called Speakeasy. This is similar to Spark, but it essentially also controls for partial correlation between the features, so it's getting an even more parsimonious explanation of all of the correlation structure because it's able to do that. So it's going to give you even more sparse representations of correlation. So in summary, compositionality can cause spurious and even opposite biological conclusions, which should be very scary to you. Dominant bugs can skew the relative abundance of the minor bugs. Even small shifts in a very dominant bug make the relative abundances of the minor bugs go up and down. Correlation is especially hard to infer, although there are some good computational tools now that use some clever tricks to get you pretty sparse predictions of the correlation network. But if you can, it's best to do the analysis with actual absolute abundances. And there are some ways to do this. So you could do qPCR using universal primers for your bugs and get the absolute abundances that way. Or you can also spike in known concentrations of foreign bugs or foreign DNA. Perhaps even do both. It would be very interesting to see a comparison of both. That's all for this video. See you next time.